Hello. Why did it move over there? Let's put you back. There we go. All right. Next question is from Barb. And Barb says, I loved your review of the two types of vaccines, the differences in each of them yesterday. Can you explain Moderna's new vaccine? Sure, fine. Yeah. And then she goes on to quote some little things from Moderna's press releases. And yep, that's absolutely right. So the um, Chinese vaccine, Sinovac, is just the entire virus with all of its proteins, but a non-working copy of the genome. So it can't actually grow. It just looks exactly like, in fact, the coronavirus. And so you are actually putting in the spike protein and all the other proteins, and so your immune system can learn to deal with these things and hopefully make a good response. And that seems to make a pretty good response. The other two types of vaccine that are being considered right now are let's see, the other one is the adenovirus delivered vaccine. So this is the Oxford one, or there are a couple other groups that seem to be doing similar things, or maybe all banded together. And this one is using whatever, a little adenovirus shell to deliver a copy of the spike gene. And it's going to be delivered in DNA form. And that DNA is then going to get turned into RNA, which will be turned into spike protein, because that's how biology works. And so we know that putting in the spike protein directly seems to be good. Putting in the spike protein only without the rest of the things and as DNA seems to be not so good. And then you've got Moderna, and Moderna's thing is that no, we're gonna break the mold. We weirdly their corporate font choice looks just like the British shopping chain Tesco. It's red letters with a blue underline underneath each letter. Yeah, weirdly. Moderna's is lowercase though, so they totally didn't bite on anybody's style. Yeah. Don't at me. Yeah, fine. Hmm. So, and Moderna's vaccine idea is also super original and novel. So they're taking not the DNA that makes the RNA that makes the protein, and not the protein. They're going right in the middle. They're saying, what if we put in the messenger RNA, which is the thing that normally comes from DNA in the one vaccine or genome RNA in the actual virus, and what if we put that in and use that to make just the spike gene? And so, there we go. Now, the thing with Moderna is, with those other two sets, we got an actual preprint that has experiments and numbers, and you can look through it with your own eyes, and you can read the words they say, which will generally be a little bit putting put that silver lining back on the cloud kind of stuff. But you can also look at the numbers and see whether or not that actually squares and compare it to a lot of different things. With Moderna, we get science by press release, and science by press release is usually kind of a thing that scientists want to avoid, but companies really, really like, because your average investor is not a biotech expert. Even your average biotech investor has probably not got a PhD in anything like that. And so they are looking at trends, and they're looking at, well, what's everybody else going to be doing, and trying to scope this out. And so something like a press release, I'm looking at it right now. It contains no data. The numbers that you see in there are the two doses that they put in, so that's okay, but we don't really know, what either of these things means. You've got a number on the drug itself, so that's all right, but that's still just a name. You've got a number of days that they gave the people the drugs for, pretty cool, and you've got a number of people, which is something really small. I thought it was like 8 people that they give this to. And let's see, one person had a low-grade adverse effect, but that's probably just something to do with the injection in general rather than the mRNA, although that's worth looking at in a study this small. And based on this, it was declared safe enough to go to phase 2. It was not declared effective enough necessarily. So what they say, the only thing they'll say, they won't say any numbers when it actually comes to the data, but they say that the amount of neutralizing antibodies, which are the kind of antibodies that can actually stick to and shut down the virus, are comparable to the amounts in people who have recovered from SARS-CoV-2. But as you know from all the papers we've covered, there is quite a range in those. And it ranges from people that basically don't make a neutralizing antibody response to people that make really high amounts of neutralizing antibody. And as scientists, we would sometimes say that anything within about tenfold means like 10 times more or 10 times less than a number is about the same, in the same ballpark, because yeah, in science it usually works that way. But with these antibodies, we're already dealing with a fairly small slice of the immune system, and it's not that big to begin with. And yeah, my worry is that they're trying to mask what I would call lackluster or small data sets. We know it's small already, but I don't know. If this was scientists talking to scientists, they would 100% be showing the data, or people would be throwing things at whoever is doing the talking, saying, "What? Yeah, is this how you think science is done right now?" So I don't know. It doesn't mean that Moderna is a bunch of, I don't know, shady or sitting on technology that is not going to work. But those are the things that it makes me think when I hear about somebody doing science by press conference. Now, hopefully, we will get something like a paper out of this very soon. Because the point of science is you do something enough that you're sure it's reproducible, and then you disseminate that, you share that with the rest of the scientific community and anybody else that wants to pick up, a journal. Yeah, fine. Yeah, it's there. It's part of the human public record. And they haven't done that yet. So I guess for now we have to reserve judgment and just say, well, when yeah, I hope they're good data when we do see them. But yeah, for now we can't see anything. So that's all I can really say about the Moderna vaccine at the moment. I'm hoping we get more on this soon. Maybe this week. I don't know. But I'll keep watching the preprint servers and news articles. Hopefully somebody picks it up. And if this thing is out somewhere and I'm just looking at the wrong spot, by all means, stick it in the comments or whatever. Send a link and I'll go read up on this thing because I'm curious. Yeah, curious but highly, highly skeptical, which is exactly what a scientist is supposed to be, right? Yeah. You're supposed to only grudgingly ever believe something when it is demonstrated just beyond any kind of reasonable doubt. When you're left with no alternative, then you say, "Ah, oh, fine, okay, I believe it." Yeah, and when it's yeah, when you're not even citing any numbers that mean anything in your press release, and you're not showing any data, then that's it's not science yet. That's all it is. It's PR. It's something designed to influence people. It seems to have influenced them. Their stock has gone up, and so good for the company, I guess. But as humanity, when do we get something out of this? I don't know. Hopefully, at some point. Thanks very much. Yeah, this has been Dr. Ben.